All right, let's jump right in. Today, we've got a fascinating explainer that honestly is going to read a lot less like your standard scientific data report and way more like a gripping biological detective story. We are going to decode the microscopic evidence of a severe localized infection, hunting for clues that are hidden entirely within raw genetic data. Together, we're unpacking the case of a microscopic suspect known to scientists as MUD-KCA23.
Okay, let's dive into this. The data we're analyzing today doesn't come from some pristine theoretical lab experiment. No, this comes from a visceral, real-world human crisis. The source metadata tells a pretty stark story. The isolation source is listed right there as a cellulitis wound swab. Specifically, this sample was taken from a human patient who is actively suffering from necrotizing cellulitis. That is a severe, rapidly progressing, and literally tissue-destroying infection. So what we're looking at today is the actual decoded genetic blueprint of the organism that was causing this devastating damage.
Tracing the timeline of this investigation reveals exactly how modern genomic forensics operates on a global scale. Just think about this. On December 16, 2025, a physical sample was collected from a patient in Benin City, Nigeria. From there, that biological material was painstakingly processed, digitally sequenced, and then submitted to the NCBI database on February 24, 2026. In just over 2 months, a physical, dangerous pathogen extracted from a hospital in West Africa was transformed into a globally accessible digital fingerprint. It's really incredible.
Part 1. Meet the suspect, Psychrobacter immobilis.
Now, what's really interesting about this slide is the sheer, staggering scale of the genetic fingerprint we're dealing with. The genome of this tiny, microscopic organism is a massive 2,923,905 base pairs long. Think of that as nearly 3 million individual levers of DNA code. And that specific code dictates exactly how this bacteria behaves, how it defends itself, and ultimately, how it attacks a human host.
To truly understand our suspect, we've got to look at its evolutionary gang affiliation, if you will, its taxonomic rap sheet. We start super broad. It's a bacteria, falling under the phylum Pseudomonadata, and specifically the class Gamma-Proteobacteria. From there, we step further down the taxonomic ladder, into the Moraxylales order, then the Moraxylaceae family, finally landing in the Psychrobacter genus. Mapping this exact taxonomy is absolutely crucial. It tells researchers exactly what family of microbes this pathogen is related to, which immediately gives us context about how it might behave and survive in the wild.
Part 2. Sequencing the evidence. The crime lab.
So, with our suspect identified, we have to ask, how exactly did researchers get from a physical wound swab to almost 3 million letters of digital code? Well, the forensic methodology here was highly meticulous. Step 1. They used an Illumina Novasec machine, which is an absolute powerhouse in modern genomic sequencing, to read the raw DNA. But they didn't just read it once. The data shows they achieved 30.0x genome coverage. That means the entire genome was effectively read 30 separate times just to ensure unquestionable accuracy. Step 2. They pieced all those overlapping reads together using assembly software called SPADES. Finally, Step 3. They ran it all through the NCBI PGAP pipeline to annotate the code, essentially highlighting exactly where the functional genes were located within that massive string of letters.
And here is what that digital crime lab uncovered. The annotation pipeline found a grand total of 2,422 genes. Of those, 2,351 are functional, protein-coding sequences. These are the actual biological machines that the bacteria builds to survive. But pay close attention to that bottom row, 26 pseudogenes. These are essentially broken pieces of genetic code. They contain frame shifts or internal stops. It literally represents the evolutionary baggage of the bacteria, tools it either doesn't use anymore or genetic mutations that just broke down along its evolutionary journey.
Part 3. The genetic arsenal, weapons, and survival gear. This structural data perfectly sets up the most critical part of our investigation. How exactly does Psychrobacter immobilis survive inside a human wound and cause such severe necrotizing damage? I mean, what specific biological weapons is it carrying? Well, to answer that, we have to sift through those 2,351 functional proteins. Hidden among all the standard cellular housekeeping genes, the genes just meant to keep the lights on, are the highly specialized tools this bacteria uses to wage war and extract resources from its host. So the crucial point is that in order to cause necrotizing cellulitis, a bacteria first has to aggressively stick to human tissue. Enter the YADA-like family protein found right here in this genome. This is a remarkably massive protein chain made up of 2,029 amino acids. In the microbial world, proteins of this specific family and immense size act as heavy-duty adherence factors. This massive molecular hook is likely what allows the bacteria to anchor itself so firmly to the host cells within the wound, totally resisting the body's attempts to flush it out.
Now, once it's firmly anchored, the bacteria has to survive the host's chemical defenses. Our genomic data shows a massive investment in nitrogen and urea processing. The core of this operation is the urease subunit alpha. This is a heavy piece of enzymatic machinery clocking in at 710 amino acids. Urease enzymes allow bacteria to break down urea, which can neutralize acidic environments and allow the bacteria to extract vital nitrogen to rapidly fuel its own growth. It is, quite literally, the ultimate survival kit for a hostile environment.
But a molecular engine that big doesn't run on its own, and this brilliantly illustrates the sheer complexity of microbial life. The main urease enzyme is actively supported by a dedicated urease accessory team, annotated here as UR-D, UR-E, UR-F, and UR-G. These 4 distinct proteins act as the mechanics. They coordinate perfectly to assemble and activate the main urease enzyme, making sure the bacteria can efficiently process nitrogen while fighting off the host's immune system.
Furthermore, to actually process urea, the bacteria has to physically pump it inside its cell wall first, right? For that, it uses a dedicated transport crew, the urea ABC transporter system. This crew includes the permease subunits URT-B and URT-C, which form the actual physical channel through the cell membrane, and that channel is powered by the ATP binding subunits URT-D and URT-E, which basically burn cellular energy to pull resources inside. It's a highly coordinated chemical logistics network happening on a microscopic scale.
Of course, a severe human wound is an incredibly volatile place. The human immune system is actively attacking. Temperatures fluctuate and chemical balances are constantly shifting. To survive all this, the bacteria relies on extreme stress responders. The annotated genome reveals specific sensor histidine kinases and response regulator transcription factors. Think of these as the bacteria's early warning radar and command center. The kinases dynamically senses external hostile changes, and then the response regulator immediately flips genetic switches to adapt the bacteria's defenses in real time. It's fascinating.
Part 4. Case closed. The bigger picture.
So we've explored the timeline, the taxonomy, the exact bioinformatics sequencing methods, and the specific microscopic weapons at play. But it's time to zoom back out from the lines of genetic code and look at the bigger picture. It's incredibly important to remember that none of this vital, life-saving data would exist without the physical, forensic work done on the ground. We absolutely have to credit the researchers responsible for this submission, KCA23, MU-KCA23, and NOEG, conducting their work at Nnamdi Azikiwe University and the Faith Mediplex Teaching Hospital in Nigeria. Their rapid isolation, sequencing, and sharing of this specific strain provide an invaluable piece of the puzzle to the global scientific community. Which leaves us with a final, pretty profound thought to consider. What does sequencing this one single bacterial genome, isolated from a severe wound in a specific city in Nigeria, actually mean for the future of global public health? Well, the reality is that every single genome we sequence and share on open platforms acts as a global early warning system. By understanding the specific genetic arsenal of strain MUD-KCA23 today, medical scientists across the entire globe are infinitely better equipped to track, understand, and ultimately defeat these devastating infectious diseases tomorrow. Thank you so much for joining me on this explainer. Keep questioning, keep learning, and I'll catch you next time.